Keith falando para nós aqui na nossa celebração de 75 anos do Instituto de Biofísica, Carlos Chagas Filho. So it is a great pleasure to have Dr. Keith talking to us as part of the program idealized by the Environmental Sciences program and in celebration of our 75th anniversary of the Institute. So this is the third and last conference by the program of Environmental Sciences. As you know, by the invitation messages, there is also a mini symposium that was going on through now, and you can also request those certificates. It was such a great set of lectures, and I'm sure that today is not going to be any different. So I pass the word to Dr. João, and thank you again for organizing and idealizing those series of talks. And thank you again, Dr. Keith, for accepting the invitation and talking to us today. Thank you.
So my pleasure to say these few words in Portuguese, so Keith will not understand. What's important here is, before everything, é um cientista ambiental especializado na análise e destino dos efeitos dos contaminantes. Obteve seu diploma em Química, Engenharia Química e depois o mestrado em Engenharia Ambiental. No doutorado, em Berkeley, trabalhou com Robert Risebrough, um dos papas da história do DDT. E eu acho que, dito isso, é uma honra enorme para o programa ter um cientista que já nos ajudou no passado, orientando nossos trabalhos, junto com o próprio Robert Risebrough, há 20 e tantos anos atrás. Então, é uma figura que eu gosto muito. Ele é filho de havaianos, surfista, toca guitarra e tem um grande coração, e é isso que importa nessa nossa rede no Brasil e no mundo. Obrigado, Keith. Thank you very much. Keith, the floor is yours.
Thank you. So it's my pleasure to be able to present to you today. Welcome from Southern California. My thanks to Professors Alonso and Torres for the invitation. And yeah, let's get into it. So as environmental scientists, I'm sure we answer or have the same mission in our professional careers, and over again the same questions when in regards to water quality come up, that we dedicate our investigations and our science to. We want clean drinking water. We want to be able to recreate in our receiving waters safely, and we also want to make sure that the non-human wildlife out there are also protected. So these basic questions are the drivers for the science that we do, and in many cases the very focused investigations on contaminants that we embark on. So as a child of the 60s, I grew up in the time when environmental awareness was taking hold, at least in North America. Of course, the salient work by Rachel Carson, Silent Spring, outlining the possibilities of what uncontrolled application of synthetic organic chemicals like DDTs can do, not just to humans, but to the environment as a whole, really started this environmental awareness movement. And since I was born right about the time when Dr. Carson published her book, it was almost destiny for me to go into environmental science as something that I wanted to pursue. It took about 10 years for that awareness to really take hold in terms of concrete work and investment into making sure that the chemical environment in our receiving waters was being monitored and assessed properly. Right around 1970, 10 years later, is when regulations first appeared and monitoring programs for compounds like DDT, Pcbs, and some of the other chemicals that I'll be talking about really started to take hold. So this set the stage for career scientists like myself, like JP, like Mariana, to really give our humble contributions and determine what these chemicals can do in the environment and how we best move forward. So that again gave me the inspiration to pursue a career in research on synthetic organic chemicals and their occurrence and impacts in the aquatic environment. The talk, I'm going to divide this talk really into two parts of my 30 years of doing this. The first part being focused on targeted compounds, and then the second part you'll see sort of broadens the whole scope of looking for the next PCB or the next PBE out there to make sure that we don't go too far off that cliff. So I think as JP mentioned, my educational background went through UC Berkeley. Got my PhD in environmental science and engineering there. I then spent 10 years as faculty at the Skidaway Institute of Oceanography in Savannah, Georgia, where I spent sort of my formative research years. And then the later half of my career, I migrated back to Southern California, and I worked at a water quality agency, research agency known as SQUIRP. So a nice division, about 10 to 15 years each place. And so I'm going to share with you some of the highlights of that work. Throughout my career, I've always been an applied researcher. I was never very interested in extremely focused fundamental research. And so the applied nature of my work lent itself to collaborating across many disciplines. And so the topics that I'll be talking about, accumulation and trophic transfer, of course, I need to work with people that understand the biology better than I do. And so people like JP and Mari come in there. I had worked at Skidaway on remedial treatments of these compounds in natural settings, marsh sediments. And so working with microbiologists and engineers was a big part of that. And then I also had an interest in how these contaminants might move around both on short spatial scales and longer transport scales. And so working with the physics side and atmospheric scientists was also a very important part of my career. Not to mention the different chemists that I've worked with. I'm typical. My background is in analytical and environmental organic chemistry. I've worked with natural products chemists and physical chemists, and so each specialty would lend something to the overall picture, and just to enrich and make the research more comprehensive. Speaking of those collaborations, I'm happy to have experienced a number of really high-level, top-notch collaborators and colleagues over the years. Again, engineers, physiologists, endocrinologists. The top picture is a panel of experts that I'll refer to later for the state of California in my later stages that I worked with very closely. The guy in the lower right-hand corner with the guitar is Dr. Walter Vetter. He's a very close colleague and friend in Germany. I came across Walter in several aspects of working on toxphene, which I'll talk about a little bit, and also natural products, halogenated compounds that are found in the environment. And then those two on the very bottom, you guys know those two people well. I think I met them. I met JP, I think, in the late 90s, and then Mari came along as one of his students, and I think that was at a dioxin conference in Australia. So collaborating with a wide range of folks is essential to my career. I would have it no other way. The other thing that I'll note is that not only working with academics and government researchers, but getting the entire picture of collaboration, to me as an applied researcher, is essential, vital to doing good work. At my position in SQUIRP, our member agencies are basically our board of directors represented treatment works people, so the very large wastewater and water recycling plant operators, as well as the regulators, so California EPA, who manage these facilities. So you get a very broad-based view and different perspectives to frame your research, I think, is a key aspect. Okay, so the first half of my career, as I mentioned, I got my PhD at UC Berkeley, and the gentleman here in the very bottom, kind of a grainy picture, but that is Dr. Robert Riceborough, very well renowned for his work on the early impacts of DDT on fish-eating birds. Bob was basically my major professor at Berkeley, and it gave me the cornerstone to do the work that I did for over 30 years. After I graduated from Berkeley, I got my first position at the Skidaway Institute of Oceanography, and my two mentors there were Dr. Richard Lee and Herb Wyndham here. Herb was the director, a trace metals guy, and then Dick Lee and I worked closely together on things like PAHs and oil spills. What that did, it allowed me to set up shop in my lab at the Skidaway Institute as a junior faculty. Here are some of my early lab staff. And I'm going to talk about two basic categories of persistent organic pollutants that did a fair amount of work over my 10 years at Skidaway. But just to give everybody this common frame of reference, my basic tool for the trade is gas chromatography coupled to mass spectrometry. Just a very quick overview of GCMS 101. To separate organic compounds in a complex environmental mixture, we really need an advanced tool or a powerful tool to do that. The first dimension would be separation of the actual components based on their volatility, and that's where the GC part comes in. You can chromatograph these compounds, separate them, and then blast them into the second part of the instrument, which is the mass spec part, which identifies, based on the molecular masses and the fragments formed, what actual compound is present in that peak at that time. There are two different modes that we use for environmental analysis. Most frequently, I think the most frequently used is on the top here. This stands for electron ionization. And basically, you use a thermal electron, a fairly high energy electron. You target it at that molecule as it comes out of the GC, and it blows it apart into different mass fragments, which is what you're seeing in the blue plot here. And you get several fragments, but the sum total of this fragmentation pattern allows you to identify with pretty good certainty what that compound is. The other mode that we use, and then the one that I'll probably be talking about a little bit more, is known as CI, or chemical ionization. This is a lower energy where you bleed in a reagent gas into your mass spec, and it imparts a softer or lower energy ionization, which preserves the compound in a more complete state, as indicated by these large lines here. You can see most of the energy here. You get many, many fragments. And if you think of scanning these different masses, you can get a lot more sensitivity if you're only scanning this narrow molecular range than you would if you had to scan this larger mass range here. So CI is a very important tool when it comes to analyzing for halogenated compounds in particular. So the first class of legacy POPs that we came across in my time at Skidaway are PCBs. The basic summary of PCBs, for those of you who aren't really familiar, is a synthetic mixture based on this two aromatic ring structure, a biphenyl, and then chlorines at any one of these 10 positions around the two rings. Because it was a mixture, theoretically, there's 209 of these compounds possible, but in reality, there are likely dozens of these compounds in an actual mixture. The range of chlorine content ranges from 1 all the way to 10. So an average of 50% would be probably 50% chlorine would be a good estimate. And because of this arrangement, these molecules are very stable. That's why they were designed for high voltage electric equipment. But some of these individual compounds were more stable than others. And so we'll see some selective weathering of that in some of the next slides. We use a whole lot of it, obviously, and of that, some of that bled out into our aquatic environments, generating obviously detectable levels of PCB contamination. And then the somewhat question of, it's a stable compound, but what about its toxicity? Still pretty controversial. People worry about levels or low-level exposure to PCBs and what it might cause in both human and wildlife vectors. But suffice to say that there is biological activity due to PCBs that we know about. So I mentioned selective weathering. These complex mixtures are not well preserved in the environment. What happens is that certain congeners will persist based on their substitution pattern and their stability. Some will transform, whether it's by biological or physical processes will bleed away. Natural attenuation will degrade them. But we end up with patterns where we can expect, like I said, probably a couple dozen to 3 dozen, 50 compounds that we can expect to find in any given environmental sample. What's unique about my time in Georgia, though, is about an hour from my institute in Savannah was an industrial estuary called the Turtle Brunswick River Estuary. And they had a large chemical plant there, a chloralkali plant, that used a very, very unique PCB mixture in their process. And as a fisherman, I was interested in analyzing fish to see how much they took up of this stuff. And the red trace here is the simplified pattern of these very highly chlorinated PCB congeners. You can see they have 8, 9, and even the decachlorobiphenyl here. A much simpler pattern than what we would see sort of worldwide as a PCB pattern. So this got us thinking, hey, if it's in the fish in this estuary, what about the apex predators that are living in that estuary? And the coastal bottlenose dolphins was our prime example of that apex predator. My graduate student at the time, Aaron Palestra, was interested in marine mammal and trophic transfer, so we set about to look at how much of this PCB Aerochlor 1268 made it to the top of the food web. And the short answer is the levels of PCBs shown in the pink highest bar here exceeded about 100 ppm on a lipid basis, so well above these fuzzy thresholds that people worry about for marine mammal health. And so this caught the attention of our National Marine Fisheries Services people that were interested in marine mammal health, Lori Schwacky and her group in Charleston, who set out to do a bunch of health assessments post publication of this work by Aaron and myself. So we felt pretty good that, at least we found a PCB mixture. We were able to document its levels and then hand off some of this occurrence and trophic transfer research to those who were qualified to do detailed health assessments. But the question that we could ask about, well, what does local contamination mean for the regional aquatic coastal ecosystem? And so we set about to see just how far north and south along the coastline that this Aerochlor 68 would bleed out or make it out of that single estuary. So Savannah was my home base, again, about 150 kilometers to the north of where the hot area was, and I'll put these circles up. The Brunswick area here is where the chemical plant was. Savannah is again to the north, And then Jacksonville is about 100 kilometers to the south across the Florida border. So we got our hold of fish from each of these three areas, and then Aaron was able to parse out the PCB profiles using PCA to show that the Savannah fish were definitely different than the Brunswick fish, and largely based on that AirCore 1268 difference. And then Jacksonville had some mixture in between. And so this led us to believe that most of the migration or transport of that 1268 was to the south and not to the north. So that's the 1268 story. Turns out right across the estuary in that same Turtle Brunswick system, Hercules Chemical, now known as Ashland, produced much of the U.S. production of another pesticide known as toxaphene. Toxaphene was also very highly chlorinated, a little bit skeleton here, not an aromatic structure, but a hydrocarbon, what they call a cyclic a hydrocarbon cycle, C10, with any number of chlorines and hanging off these substitution positions. The way that toxaphene was produced was very non-specific. They basically took pine trees, extracted the pine resin, distilled the resin to form this compound called pinene. You might be familiar with pine sol, these cleaning agents. That's what that is. It smells like lemon or pine oil. And then they just basically chlorinated the heck out of that and came up with a pretty complex mixture, which I'm showing here on the bottom. If you chromatograph technical toxaphene, you get hundreds of peaks, and in those hundreds of peaks, you probably have 2 or 3 or more compounds co-eluting under that mess. So a very complex mixture to analyze with GCMS. But the bigger question is, okay, once you put that into the environment, what happens to it? And we know from work by people like Terry Beidelman that, hey, this stuff, and Gary Stern in Canada, this stuff will also transform, losing chlorines for the most part, to give us a different mixture. And this is a weathered profile at the top. Certain compounds stick out. This hexachlorinated bornane showed up very prominently, as well as this heptachlorinated bornane in samples over and over again from that estuary. So going from the original mixture to the weathered mixture gave us our first insight to how we attack or analyze for this compound. The state-of-the-art at the time, the one that regulators and that most commercial labs would use would be based on electron capture detection, or ECD. The problem with ECD is that it can pick up any number of halogenated or even non-halogenated substances that were amenable to electron capture. Even sulfur would light up the detector. So you could get peaks, but you weren't positive that they were toxaphene, or you weren't positive that they were PCBs. And in an estuary where you had both, that generated some uncertainty as to what you were detecting and quantifying. So when we got involved, one of my colleagues at Skidaway, Stu Wakeham, had an old mass spec that he loved and took home with him, and probably took to bed with him. Don't tell him I said that. But his instrument allowed us to do the first chemical ionization work for toxaphene. So we applied that NCI method that I talked about earlier that is sensitive for organohalogens, and we were able to positively identify those prominent chlorinated bornane compounds from the PCBs. But we went on to do some work with Fungulus and how it eliminates these residues. We worked with the local governments and the EPA to monitor the recovery of that marsh system. They actually excavated or took out some of the more heavily contaminated sediments, and so we did pre and post fish collection to see whether toxaphene levels declined, which they in fact did. And so this is some of the work that we were able to do after we fine-tuned and developed that NCI method. It ended up that that old ECD method that people had used and basically turned out a bunch of uncertain and really pretty much worthless data. EPA updated the method to include the negative chemical ionization in a standard method that's now published in their analytical Bible, which is known as STW846. It includes these residue congeners as well as original congeners, so that's good. But one of the remaining issues is that, especially in places like that where you've got co-occurring organochlorines such as Pcbs, you're still going to get some possible interference. And the way this works is the blue are Pcb masses and the reds are indicative of these toxaphene congeners. But in the mass spec itself, you could get some interaction where some of these ions could end up in this cluster here. And I won't get into that, but it is possible based on oxygen contamination in your instrument. And so some ways around that we found are you could use a brute force method ahead of the analysis to pre-separate out the Pcbs. The Pcbs being slightly more nonpolar, we can wash them down with hexane from a column of the extract and throw that away or analyze that fraction separately. And then we can retain the chlorobornanes in a second, slightly less polar or slightly more polar fraction and analyze that using the 8276 method. So a brute force method to circumvent this PCB interference problem, and which is real. We actually took two samples. One was a halibut or flatfish sample from the fish market from Alaska, which we would expect to have low levels of all this stuff. And then a sample of fish actually from Southern California that we knew had high PCBs. And so we applied the method with and without accounting for these co-occurring interferences. And you can see if the levels of PCBs are low, the estimates of toxaphene are really unchanged. But if you've got high PCB interference, the potential for misrepresenting the toxaphene residue concentration is pretty large. It's a 10-fold difference, non-subtracted versus subtracted. So you want to be aware of this when you're working in contaminated systems like that. Okay, so much for that first part of my career. About 2005, I got homesick and I moved back to Southern California, and I ended up at a place known as SQUIRP. And SQUIRP is unique in that it's not an academic, it's not a real government agency. We're sort of a pseudo government agency, but we do water quality for larger issues as well as regional issues. What happened when I got to SQUIRP back in that 2005 timeframe is legacies were basically on their way out. We knew a lot about them. In some cases, we could do something about it. And in other cases, it was better to just leave Mother Nature to slowly degrade these things away and watch their levels decline. PBDs is a classic example of when I first got there. We did some work on that. They were elevated, but have since been banned, and now their levels are declining. But what happened when I got to SQUIRP and started working with different folks? This is Alvina Mahinto. She's was sort of my right-hand person during the later parts of my career at SQUIRP. She's a molecular toxicologist by training, and so our complementary backgrounds will lead us down this path that I'll show in a couple of seconds here. We focused on, hey, let's forget about the legacy stuff for now. What's coming down the road? What about all these endocrine disruptors? What about newly registered pesticides? How can we screen for these things when there's so many of them? And that's what's indicated here. You know, there are thousands and thousands of chemicals that we never ever even analyzed for. And basically what happens is they create a chemical mixture or a soup here, a complex soup. And so the real question is, what does this soup do in terms of impact to our receiving waters? Not an easy question. A lot of people would say just focus on individual things like perfluorinated or whatever the contaminant du jour is at the present time. But we wanted to take a little more holistic, broader approach. And so the state of California and I got involved from SQUIRP in bringing together a panel of experts to give us a more comprehensive approach than just this chemical by chemical assessment. And so their recommendation basically boiled down to, we need this funnel we need a better funnel. Our funnel right now is too slow and too limited. So what can we do to broaden the universe that this funnel does, and how do we develop these tools that give us the right products at the end? And basically came up with 2 recommendations. You need to use advances in bioanalytical measurement to help guide this funneling process, and then non-targeted as opposed to focusing on single classes of contaminants. So the first tool, the bioanalytical approach, is basically applying these cell lines that are engineered to respond to classes of chemicals by mode of action. So not solely based on their individual structure, but how they make that genetic material respond. And I'll use estrogenicity as an example here. If you've got estrogenic compounds that want to feminize, say, fish, when they interact with this engineered cell line, they're going to react with the machinery that activates the expression of a protein, like an egg protein, the telogenin. And that linkage there, or that response, is what we want to capture as an integrated measure of how these estrogens might activate this adverse outcome of feminization of fish. And it turns out that there are commercial cell lines where you can then translate that response into an equivalent concentration using a reference toxicant, E2 being 17-beta estradiol. So that was a huge leap for us, not just a response that could not be quantified, but an actual concentration that we can start to think about setting benchmarks and thresholds. So we with our team, we set about building a toolbox that had many of these tools, estrogenicity just being one endpoint of interest, and to see if we could make this bioscreening approach work from a receiving water standpoint. A number of steps that have to be taken through to spit out a successful test endpoint. And I don't have time to go into all these in detail, but suffice to say that each one of these steps is a very important part of the process. We want to make sure that these cell lines are sensitive enough to see compounds at their impact level in the environment. And as we know from the literature, estrogens can be bioactive at very low levels, at the part per trillion or nanogram per liter range. So we want to make sure these cells can respond to that, and we can select the right ones based on that criteria. Very important then to take the good candidates and make sure we have a well-controlled standardized protocol, just like we do with analytical chemistry. If we can't reproduce these results or make them comparable across labs, we failed. And so a good amount of our time was spent standardizing these protocols, incorporating quality assurance criteria. Just as important for analytical chemistry, this is the same approach, just using cells. And so we use the same kind of criteria we would use for mass spec as a sensor, including now that we have living cells as our sensor, making sure that these cells are happy enough to respond in the way we want them to do. So once we have standardized protocols, we wanted to make sure that we could see responses, that these tools were actually capable of seeing differences among samples. So what I'm showing here is a bunch of samples. One would be the effluent from a typical municipal wastewater treatment plant. So it had some activity or some contaminants left. And then going through a purification process for recycled water, you can see here, these things are going and increasing water quality, and your final recycled water product you would expect to be very clean. And that's what we saw when we applied these assays. We saw a stair-stepping down of activity of estrogenicity in these different samples. And not only that, we showed that 4 different labs can basically get the same trend. So we're on our way to showing that this approach is feasible. Okay, so quickly on to the next tool, non-target analysis. Back to the mass spec side of things. Non-targeted is basically doing a broad scan as opposed to focusing on a certain group of ions, which means that it's a lot more work and you generate a lot more data and you generate these complex, now two-dimensional heat maps of stuff that would be chromatographed in any given sample. Very complex process, but the possibilities for applications are pretty strong here. I'm going to focus on fingerprinting as the first application because it doesn't involve, it's sort of the intermediate complexity workflow where you can take heat maps instead of drilling down and trying to identify one little frame of that heat map, and that's a lot more work. Fingerprinting, and so working with Yunha Ho at San Diego State and her students, she used a two-dimensional GC and with a time of flight mass spec to look for accumulative or persistent compounds that aren't picked up by those targeted PCB and toxicity analyses. And she had a series of students that looked at marine mammal or dolphin blubber off the Southern California coast. She found that there are DDT-like compounds that not necessarily originate from the DDT technical mixture, but from other related formulations. You might have heard of tris or dicafol. So dozens of these kinds of compounds that we don't normally see were discovered in these samples. We looked at different species of marine mammals to see whether we could to see whether there are differences in their fingerprints. So in other words, whether one species would be the ultimate sentinel and others would knock down the signal or have less contamination. And that was Kasaboon's work most recently. We also applied Yunha's instrument to non-biological matrices. Stormwater runoff, as you know, can be a source of contaminants into the coastal ocean. And we saw here that different land uses, the urban parking lots versus agricultural land versus open space, clearly we could distinguish these non-targeted fingerprints based on land use. So again, we're on our way to showing that this tool could be useful for fingerprinting. Nellie Shaw was another one of Yunha's students who looked at in detail at coastal and offshore bottlenose dolphins, working with our colleagues at the Marine Fishery Services in San Diego, California. And basically her non-targeted work showed that, 24 different compound classes are detectable using non-targeted. You got your DDTs, you got your tris compounds, you got PBDs. And then you also pick up, again, these natural compounds. They're not all synthetic organic halogens, but natural compounds come into play too. And then again, of course, Mariana came to visit us to do some postdoc work, bringing along samples from your neck of the woods down along the Rio coast. Same species, coastal bottlenose dolphins. And although many of the compounds that Mari found were similar, some stuck out as being different. And I just circled a couple of them. There appears to be some legacy Myrex that is very prominent down in your area, whereas up in Southern California, we didn't see that. And so that enabled Mari to do some statistical comparisons, looking at the signatures to see if they were distinctly different. And she concluded that, of course, we can use PCA to show that these separate out and that signatures could be used to look at geographical influences. The other thing that Nellie, again, did was half her samples were of an offshore. We have offshore islands here up the Southern California coast, about anywhere from 20 to 60 miles or 40 to 100 kilometers. And then we have those that hug our beaches and shorelines. I know Mari's probably seen those while she was surfing. And we asked the question whether they can have different non-targeted profiles. And looking at natural products, we concluded that the answer was yes, we can distinguish between these animals based on their complex profile of these naturally halogenated compounds. Lastly, the non-targeted work still is again very complex and is does not always identify exactly what's there. So a number of tentatively identified compounds, as well as completely still unidentified compounds, emerge from these types of analysis, which gives us, some questions to ask in future research. Yeah. So that's the science in 30 minutes of my career, my life in 30 minutes. My observations over that time, again, born in the early 60s, when if we jumped in the ocean, we'd likely be swimming in visible sewage residue to now when I go out fishing and the water is blue and for the most part very clear. So from my very humble perspective, we've come a long way. We've removed a lot of this gross contamination, and the way we are now, the present, you can tell by the compounds that we focus on now, the perfluorinateds, the microplastics and whatnot, that we're looking at parts per trillion levels now. So clearly we're focusing down. And I'm not saying that's a bad thing, but we just have to keep that, again, in perspective. I think we do need to remain vigilant and make sure that another future PCB doesn't pop out. We can look at other things that are maybe more prevalent now, or at least more attention is being paid to as a result of human activity. But the question remains, am I going to ride off into the sunset and forget about this stuff? I think the answer is no. I think I'm more like the little baby here. I think we need to keep our eyes peeled, again, be vigilant to make sure that things are continue to improve as opposed to deteriorate. And one way to do that, I think, is to look at these tools that we just talked about. And I'm going to go straight to this figure. I think these bioanalytical tools and this non-targeted analysis do have a role in the current risk assessment for chemicals. I think the cell assays will broaden the scope of screening and then allow us to focus in on the bioactive compounds as opposed to the compounds that don't elicit responses that we don't care about. And this is very important in our world of dollars today, right? We can go to the field. We know field work is very expensive. We can do bench scale testing and non-targeted, again, which is very expensive. But we need relatively quick and inexpensive screening tools to guide the dollars that we spend later on when we think we have an issue. So with that, I'm going to stop and say, hey, remain vigilant. You guys, the future is bright, and I think the questions asked will change, but the scientific process will not. I encourage you all to hang on to those great collaborations. There are everybody experiences not so great collaborations, one-way collaborations, but when you find people that are a pleasure to work with and that you can contribute to collectively as a team, you got to hang on to those. And then keep an open mind, understand that you are just one cog in the wheel, understand your role and your contributions, as little as it may seem, they are important. And with that, I'll just say enjoy the view and thank you for your attention.
Thank you very much, Dr. Keith. It's really, really interesting talk. I think we can open for questions and comments right now. And I'm not sure, João, do you want to talk a little bit while I read the questions here?
Just to say thank you, Keith. Thank you very much. This very important view, and we know for sure that if we had all clean, we still have other work to do.
Definitely.
Thank you so much.
Yep, no problem.
Yeah, it's true. I think we are always going to be using and disposing of something that shouldn't go where they're going. It's really nice, and I think it's a great message to keep looking and be diverse, and especially what you said, as look for what makes the worst impact and try to be the most efficient on cleaning up, or better yet, not disposing of it in the wrong way, but cleaning it up as well. Mariana, do you want to make a comment before I start with the comments?
I would like to say thank you very much for Keith accept our invitation.
Oh, you're welcome.
So Keith has so many good works that is hard to show in just 30 minutes, but you did a great job. And we see there's a lot of emerging contaminants that we can still looking for in Brazil, in California, and everywhere. So it's hard, but we have to do this dirty work. And I would like if you could talk a little bit more about the natural products, my favorite ones.
Sure.
Thank you.
No problem. Let me try to open another one. I had some backup slides on a different one. Oops. So Mariana's questions was, well, what about these natural products that you see? And she certainly saw many of them in her samples using the non-targeted, and everybody else does as well. So the tools that we use allows us to see these naturally produced compounds. Again, the mass spec will not discriminate whether it's a synthetic or a natural compound. So there are halogenated compounds, especially in the marine environment, that pop up in environmental samples. And the real question is, okay, will these have a different biological effect or toxicological effect than their synthetic analogs? I think that's probably one of the questions that we've been focused on for a while. And based on their structures, the answer would be, well, I don't see why it would be fundamentally different. Again, you see a tribromoindole here that we actually found by accident in oysters off the Georgia coast. We had a big storm, a hurricane come in, and then we were out sampling about a week or two after the hurricane passed. And the only peaks we saw were these three big peaks, and then they didn't correspond to anything like Pcbs or chlorines or anything like that. It turned out that they were these natural compounds. And so, what's the role of these compounds? There's a whole different world, a different community of researchers that look at this stuff. Many of these compounds are actually transformation products or breakdown products of much more complex compounds that are secreted by seaweeds or bacteria that live on corals, and so stuff like this. So this led us to start looking at reef ecosystems. This was a hard bottom reef off the Georgia coast, and so we started to analyze stuff like this, and we found a number of these compounds. I didn't have the toxicological connections back then that I do now. And I'm hoping people like Mariana and others will take this work and determine whether the bioactivity of these compounds are similar, lower, higher, the same as what we see from synthetics. And then address the question, okay, the total contaminant load in these marine mammals, what does that mean? Do we concern ourselves more with these compounds or less? So Marty, I'm hoping you and your colleagues will take up where us old geezers have left off and start addressing these larger questions.
Okay, we will try. We will invite you to do the research with us. Thank you.
Yeah, and you need to contact Alvina. That was my last message to you. If you need me to get involved. So I am retired from Squirt, but I'm still active a little bit scientifically. I'm a editor for Chemosphere, so I'm hoping to stay in the science. So you and Miriam and JP and anybody listening, if you have any curiosity or questions, or you're looking for connections in our part of the world, feel free to contact me. No worries.
Sure. Thank you so much.
Thank you.
Want to come back here, Maria? Are you looking to come back to Southern California at some point?
Sure. Why not?
You teach your baby to surf?
Yes.
You can teach them to surf. I'll teach them how to fish.
So I think I'll open to, there are a lot of comments on the YouTube, thanking you for the talk, saying it was really great and opening eyes, and it was really, really good. There is a question from Bruce, a professor from the Institute. He asks, what I read was that if it was difficult to contaminate the groundwater table with organic contaminants. Is that true? And if true, what is the reason for that difficulty? If you could also comment on the aquifers, if it has a comparable behavior to the groundwater table.
That's probably a question I can't answer in detail. I'm certainly not a groundwater transporter hydrologist. What I will say is that clearly it depends on the situation of the soil and the aquifer. Some aquifers probably have easier transmissivity than others. I think for the most part, you will definitely get attenuation of contamination as you work your way down the soil. So the best working aquifer or the most highly filtering soils will attenuate that, and so you will get low residuals. But there are certainly cases where groundwater contamination is an issue. Shallow water tables where the distance traveled is shorter. In those cases, you will see issues with contaminant. I think the perfluorinated compounds is a classic example of that, where people are very concerned about trace levels showing up in their groundwater. I don't have, again, a lot of practical experience with groundwater contamination, so that's probably about all I can say about that.
Okay, thank you. And I have one question. I guess there is a continuation to the question that do microorganisms in between have a fundamental role?
I'm sorry, microorganisms, do they?
Yes, if they have a fundamental role on this.
I think they have a very important role. I'll start with the natural products. I think the microorganisms or the bacterial consortia that are on corals are probably a very key consortium or play a very big role in transforming these large halogenated compounds and breaking them down into the compounds that we can detect. Whether that, and then what role that has on their symbionts, the fish that feed on these corals, there's a lot of literature showing that these halogenated natural compounds are feeding deterrents, or it's chemical warfare against fish feeding on the corals or their growth. So the microbes associated with that film on the corals is very important. Certainly, microbes are important in sediment ecosystems and natural ecosystems where we need transformation of PCBs, toxaphene, any other contaminants coming down the road. We need that to happen to get rid of that persistency. Some of the secondary questions of that is, okay, are we increasing the toxicity by transforming these things, or are we decreasing, or is it neutral? We certainly increase the mobility of these compounds. I'll use DDT as an example. As we know, 4,4-DDE, the main product from DDT, is very persistent. But over time, DDT will degrade into lower chlorinated compounds DDMU with, and whatnot. These things are more water soluble, they're more volatile, so you have increased mobility once you degrade this. And that acts to disperse the overall contamination from the area of interest. But what that means for the areas that receive this, again, is a question. But basically, you're allowing a wider dispersal and dilution of this stuff as you degrade these compounds. And microbes play a very important role in that.
I have a quick question. I'm not sure if it is a concern or not. With the thawing of the ice caps and this maybe raise in the water level, do you see or can you see different compounds being found that would be natural, but they were trapped in those frozen areas, and that now might change the ecosystem or might change the
right. So very appropriate and great question. I personally haven't done any research on climate change. I think I'm a little bit old school, and again, this issue has come about in the last few years. But that certainly is a hypothesis I think that many people are looking into, whether you're releasing not only contaminants from the immediately impact polar regions, but also as you have sea level rise, right, based on the warming, and also changes in ocean chemistry. One of the things that came up while I was still at SCWRP was ocean acidification. So not only does the volume going to increase, but the pH and the acidity of the ocean is also changing due to human influences. And so how does that affect the occurrence and the toxicity of these, especially these ionizable compounds? Okay? Neutral compounds like the Pcbs, slight changes in pH probably won't have much of an effect. And sea level rise, again, unless you're inundating former waste plants or dumps along the coast, the release is probably again going to be muted. But the newer contaminants, the ones that are more affected by slight subtle changes in water chemistry, certainly can be thought of as changing as a result of climate change. So I think this is a wide open field for you younger scientists to really latch on to. So great question. I can only speculate very generally on maybe some of the topics to look into.
Okay. And I guess one last comment. How do you think that like an explosion like that in Beirut might impact with the cloud of chemicals probably are going to be falling down as rain or probably as sediment and impacting also, like, do you think that there might be some acute changes there that can disperse, or do you think that something like that can be more easily cleared with the technology that we have now, or at least monitored such that it might not have such an impact?
The explosion you're referring to is, sorry, I missed the first part.
Oh, sorry, the explosion of that warehouse or that happened that probably had all the chemicals being like that mushroom, the cloud that came down, and probably that will come down and contaminate both soil and water, and that might all fall down into the ocean at the end. Do you think that we have techniques to monitor that such that it can have a smaller impact than that would have a few years back, or we still will have to wait and just see what happens?
Yeah, that's a great question. I think what I can, the only way I can answer that is I'm going to use an equivalent the event, the response to these large oil spill blowouts. So I'll use two examples, right? The first example is the Exxon Valdez spill in 1989, right, where a ship broke apart and the oil came out. So started with a local spill. Obviously, the response time in cleaning up or monitoring this oil spill was not so great, and the oil spill spread and creating a pretty widespread impact on the Alaskan coast. The second one was the Deepwater Horizon platform blowout in the Gulf of Mexico, which occurred very deep in the ocean. So I'll just turn your atmospheric explosion question upside down and think of a deep water explosion as, you know, being an inverted atmosphere. And there, again, the access was very difficult to get to do any sort of on-site remedial actions. What I know they did do was they responded by dropping in dispersant to lessen the clumping or aggregation of the oil as it came up to the surface and spread out toward the coastline. So that's something that, again, is still controversial. I think it's in all plans to try this when you have nothing better to fall back. The default, the option was to just let it spread. I mean, that was the technology. Collect it with physical booms and use a dispersant. So again, as a feeder or inspiration to the younger generation, maybe we need a third option. Maybe we need 4 options. And that's where, you know, you guys come in as, hey, you know, come up with that idea and see it through. See if you can come up with a third option to either treat or monitor that situation in a quicker, better way, giving you more information to judge your actions on. And again, so I'll end the answer to this question by most of the research we did was to inform the people that make the decisions. Hey, here's the science. Now it's your responsibility to make the right decision based on that science. So as scientists, all we can do is provide that good science or that good technology to allow these decision makers to make the right move.
Thank you. Thank you very much. I think there is another comment, question by Bruce here. If there is time, I have an additional question. How is you with time? Keith, is that okay?
Yes, I saw the question. Yeah, I'm fine. Is this the microphone?
Yes.
So I'll make this answer very short. I think it's a waste of time. You know,
okay.
Again, being born in the 60s, you guys, and I'm not saying that I'm right, okay, but based on what I know and what I've seen no science to suggest that there is really an impact where other than physical, you know, choking of larger debris. Clearly, that's a problem in the ocean. Large debris, the Pacific garbage patch, that's despicable. Okay, so what I'm referring to now are small plastic particles that we're trying to measure and that people are doing tox studies on. Growing up, we have McDonald's, okay? And growing up, I drank my fair share of McDonald's shakes, you know, the ice cream beverage, okay? Now, in McDonald's shakes, it became known to me that they include a synthetic thickener in these shakes. It's a silica particle. So it's basically like very fine sand. Okay? They use that to thicken things, okay? So I've ingested probably lots of sand in my time. Okay? So I'm equating these plastics to the sand. Okay? By itself, I can't see a credible way where, you know, again, unless you're ingesting shakes for the rest of your life as your only liquid, I can't see where that would be a problem. Now, if things are attached to the shake or the particles, you know, like gobs of PCBs or gobs of pharmaceutical or something like that, then yeah, you have a credible exposure route. But the plastic itself is pretty inert. I'm a chemical engineer. PVC, polyethylene, these things are fairly inert. You're not going to be exposed to the monomer, you know, that's used to generate these plastics, again, unless some reversible reaction happens with it. So I think it's one of these public perception deals where people, you know, you can detect it, we know it's there, and now people want to know what the heck you're going to do with it. And we have to educate the press not to completely sensationalize that's my take. Sorry, longer than I wanted.
No, but that's good. It's always good. Thank you very much. So there's a lot of other thanks here from the immunobiology program, thanking both you and the environmental sciences program for the amazing talks. All of them were really great. And so I think I can now pass the word to João Paulo to Mariana. I thank you again. Keith, it was really amazing talk, amazing discussion. Thank you very much.
Well, thank you for hosting, and Madi and JP, thank you. It's good to see you guys on the screen, and I hope to see you guys sometime in the future, in person.
That's it. Our feeling is more or less the same. We have a lot of good ideas, and the importance is someone has to follow and to create over our shoulders. So we will try to maintain the university and the courses and everything for them. They are the real that will make the difference one day. Thank you very much, Keith.
Yeah, thank you guys. Again, thanks for the opportunity and invitation.
Keith, my last question.
Wow, I'm getting grilled. Yeah, go ahead.
What do you think will be the next group of emerging contaminants?
Wow, if I knew the answer to that, Mariana, I probably wouldn't have retired. But I couldn't speculate. I'll admit that in my 9 months of retirement, I haven't really spent too much time worrying about that. So I can't give you any guidance. I think that I'll use perfluoros as an example of, it's there. The guidelines that we're setting, and I don't know what the situation in Brazil is like for perfluoros, but in the States and North America, a lot of effort and a lot of resources are being used to look at these, nanogram per level monitoring levels. And so I'm not saying that that's misdirected, but I think at some point that will go away, just because, again, they stop using the chemical. I think that's the key, right? Is when we know levels are stabilized and decreasing, then we know we've done a good management action. If it's the ones that keep going up that we have to worry about. So I will answer your question. Look for the replacement chemicals. Those are the ones that I think deserve attention, right? And so one example of a replacement chemical is these organo phosphate flame retardants, right?
Mm-hmm. Yeah.
When 5 years ago we saw those levels were the highest of anything we saw in these broad scan surveys. So I think they've gone away from chlorinated TCEP, TDC, PP. I think they've shied away from using those, but now they're still using the non-chlorinated versions. And so I think that class of organophosphate retardants is something worthwhile looking at. The other class of chemicals that we always are concerned about are biocides or pesticides, because they're meant to be bioactive and selectively target pests. So the new generation pesticides, I think, are always something to keep an eye on. Okay, I know you worked on a little bit on, what was it, pyrethroids, or what was the other one? Can't think of it now. Oh, well, neonicotinoids are, that was in vogue for the last decade. I think that's probably died out. I think the bees were one specific target, but I don't think they've seen any broad spectrum neonics. I could be wrong. But pesticides, replacement chemicals, I think that's where focus should be made on. And it kind of all falls under this umbrella of green chemistry, right? We need to be smarter about what we replace banned chemicals with. We shouldn't pick something that's just as bad or worse, obviously, than what we're replacing.
And the personal care products also, or not?
Yeah, you want me to say UV filters, don't you?
The conservatives, yeah, Sunscreens.
And sunscreen. Being fairly dark-skinned, I try to avoid sunscreen, but Mari, so think of the benefits versus the downside, right? And DDT is the same example, right? In areas where mosquito infestation is a problem with malaria, DDT apparently works. And so, how do you say you can't use that because my dolphins are being impacted? I mean, it's a difficult argument. So you must weigh the benefits as well as the downside. And I think more often than not, humans are too human-centric, and we only see the benefits to humans. Okay? So we have to take a larger picture. We all enjoy the world as a whole, so we have to weigh the good for the entire biosphere as well as just for humans.
Yeah, it's true. Okay, thank you so much again, Keith.
No problem. Good luck.
So thank you again. So now end our session here. Thank you again for the enlightening discussion and all the conversation. And I invite you to, we always have these meetings on Wednesday. So if you would like to see something different, you're welcome to join us every Wednesday at noon. Your time is really bad, right? Very early for you, but you're always invited. So I'm going to just speak in português a little bit. So, então, obrigada de novo todo mundo. Semana que vem a gente vai ter uma palestra avulsa pelo Dr. Evaldo José Palantinsky, o lado desumano de uma missão humanitária. Então, espero ver todo mundo aqui e quero fazer uma homenagem pública à Sandrinha, que está fazendo 45 anos de casa. Eu não imagino a casa sem a Sandrinha, então, obrigada de todo mundo, né, todo o corpo social agradecendo. Um prazer ter você com a gente e resolver todos os nossos problemas antes mesmo deles acontecerem. So, thank you. Thanking our secretary who is she's doing her 45th anniversary at the Institute, and she is able to resolve the problems before they even appear to us. So she's a magical one that makes everything work. So just taking this opportunity to thank her in public. So thank you again for being here, and hope to being able to talk in the future. And definitely you look like, and from what JP Mariana says, a great person to collaborate. So if I ever have anything related to water, I would definitely contact you. Thank you again.
Okay, keep up the good work, and bye-bye.
Thank you, Keith. Bye.
Obrigada, pessoal. Até semana que vem.